So these are the presenter disclosures. I will keep this up there for more than 2 milliseconds, like most people keep their slides up there for that. Okay, everybody got their pictures taken? Good. So let me first go through the opportunities and announcements, starting with our wonderful support from the Hope Foundation and recently funded awards. And these are the upcoming funding opportunities. There's a virtual grant writing workshop, which has been very well received, and applications are due May 1. NCORE pilot grants applications are coming up June 1, and these dates come up quicker than you think. We're already in the first week of April. The Hope Foundation Seed Fund soon thereafter. That deadline is July 1. And then the Impact Award deadline is also July 1 with a bit more funding. So this is all on the Hope Foundation website. Please frequent that and take a look and look at the opportunities, because there are wonderful opportunities, and most of those opportunities come from donations from everybody here in the audience and colleagues and the great work done at Hope in raising funds. We have some recent funded awards from the Hope Foundation. Elizabeth Brem is the recipient of the Charles Coltman Fellowship from UC Irvine, and you can read the title here. And Rashekar Chakrabarty, from Columbia University, has been the recipient of the Impact Award. So congratulations to you both, and we look forward to you being on the podium presenting in the future. So good luck to you all. So today's session will be quick. It's a template that I've now developed over the last couple years. It's been easy when you're sitting at your desk all the time. I do a brief introduction, then we like to highlight wonderful work supported by the Hope Foundation. Then we like to highlight the translational medicine within an individual committee. And then we have a scientific speaker. So you see the lineup for today, and I'm very pleased that this first choice for each of these categories and everyone accepted, and I think we have a nice program for today. So having said that, I would like to introduce our first speaker, Priyanka Sharma. And just a brief background on Priyanka. She's Professor of Medicine, University of Kansas Medical Center, and Assistant Director of Clinical Research and Co-Program Leader for the Drug Discovery, Development, and Experimental Therapeutics Program at the University of Kansas Cancer Center. She is the institutional PI for SWOG and also the co-PI for their NCORE grant, so she is busy. Dr. Sharma is Vice Chair of the SWOG Breast Committee and a member of the SWOG Board of Governors and a member of the NCI Breast Cancer Steering Committee. So she knows her stuff, and she will give a wonderful presentation. Thank you for joining us.
Thank you, Dr. Ellis, for the nice introduction, and I'll let you all be the judge of whether the presentation was wonderful or not. Maybe at the end of it, I'll count how many of you are still awake. But truly, this is a great honor, and I want to thank Dr. Ellis and Ray and the SWOG leadership for giving me this opportunity to present our work, which was supported by many entities, including Hope Foundation. These are my disclosures. I have them up there again. I know they were shown. What I'm going to talk about today is how we can use homologous recombination deficiency to target and treat triple-negative breast cancer. Homologous recombination deficiency, or the BRCA-like phenotype, it simply means the presence of inherited or acquired defects in homologous recombination, and this can lead to therapeutic exploitation. And to this end, targeting HRD and looking at ways to increase HRD is being investigated in many cancers, including breast cancer. So it was about 17 years ago now that seminal work from Ashworth and colleague demonstrated that BRCA1 and 2 defects combined with PARP inhibition was synthetically lethal. And then this preclinical finding successfully translated in clinics, and now we have PARP inhibitors that we use to treat our patients with BRCA mutations in many tumor types. if you look at triple-negative breast cancer, about 50 to 60% of TNBC will demonstrate homologous recombination deficiency. Only about 15% of this is being contributed by germline BRCA1 or 2 mutation, and we currently have PARP inhibitors that we can recommend for these patients in the metastatic setting. There are other combinations that are being assessed. HRD can be produced by other measures besides germline BRCA1 or 2 mutation, and our work and work by others have shown that presence of HRD is associated with sensitivity to DNA-damaging chemotherapy, and other agents like PARP inhibitors, HDAC inhibitors, and BET inhibitors are being assessed with the HRD background in triple-negative breast cancer. So with this premise, I want to talk about SWOG 1416, which is a randomized phase 2 study that assessed addition of veliparib, a PARP inhibitor, to cisplatin chemotherapy in patients with triple-negative breast cancer or those with BRCA1 or 2 associated HER2-negative breast cancer. And I want to acknowledge the work of Eve Rodler, who's the clinical PI of this study, and also support and leadership of Dr. Hayes and Hodabagi, who were the chairs for the breast TM committee and main committee to help us shepherd this through CTEP. I still remember the first set of provisors from CTEP we got on this study had over 100 comments. But with the help of the study team and our leadership, we were able to address them and move this study along. In terms of the background that led to the study design and the treatment arms, metastatic triple-negative breast cancer is associated with poor outcomes with a survival of 12 to 18 months after diagnosis, compared to over 3 plus or 5 years in other subtypes of breast cancer, so clearly an area of unmet clinical need. PARP inhibitors are more active than single-agent chemotherapy in BRCA-associated metastatic breast cancer, are not approved for those patients, and PARP inhibitor efficacy has been seen beyond BRCA1 and 2 in other cancer types like ovarian cancer and prostate cancer. And HRD assays are actually available to select patients for these modalities in clinic for ovarian cancer. However, when we look at breast cancer, PARP inhibitor efficacy beyond germline BRCA1 and 2 has not been noted in triple-negative breast cancer, and perhaps there we need combinatorial approaches and biomarker selection to really see if these agents will work beyond germline BRCA1 or 2 in triple-negative breast cancer. The challenge there has been the myelotoxicity. That has been the limiting feature when we combine PARP inhibition with chemotherapy, and the PARP trapping potency of these PARP inhibitors is tied to its myelotoxicity. Agents that have higher trapping potency are also more myelotoxic. Veliparib is a small molecule inhibitor of PARP1 and 2 with minimal PARP trapping. Thus, it can be safely combined with cisplatin at near single-agent dose. And SWOG 146 really sought to examine if addition of PARP inhibitor to cisplatin chemotherapy will be effective in the BRCA-like phenotype TNBC outside of germline BRCA1 or 2. So there are many BRCA-like or HRD biomarkers that are out there, some that we use for clinical decision-making, many that have been assessed in clinical trials. And instead of relying on just 1 or 2 biomarkers, SWOG 146 really used a multi-pronged approach and looked at multiple biomarkers to assign this BRCA-like phenotype. This is the study design. Patients with metastatic triple-negative breast cancer, or those with BRCA-associated HER2-negative breast cancer that had received 1 or less chemotherapy for their metastatic disease were randomized to receive either cisplatin with placebo or cisplatin with veliparib. The primary endpoint of the trial was progression-free survival in the 3 pre-specified subgroups: the germline BRCA mutant group, the BRCA-like group, and the non-BRCA-like group. This trial was unique that the assignment to groups happened post-randomization to allow patients to enroll and start treatment rapidly. So post-randomization testing for germline BRCA and BRCA-like biomarkers assigned patients in these groups. Germline BRCA testing was the first step in this process, and those with the deleterious or suspected deleterious mutation formed the germline BRCA group. In those without BRCA mutation, BRCA-like biomarker testing included 4 markers: HRD genomic instability score, somatic BRCA1 or 2 mutation, or BRCA1 promoter methylation, all done on primary tumor, and germline mutations in genes outside of BRCA1 and 2 using germline DNA. And a positive test on any one of those 4 markers placed a patient in the BRCA-like group, and those that tested negative for all 4 were in the non-BRCA-like group. 321 eligible patients were enrolled. We can see about 70% of patients received this treatment as first-line treatment. A very small proportion of these patients had received a prior platinum, and a very, very small fraction received immunotherapy, given the time period that this trial enrolled in. This figure here describes the distribution of patients in the predefined group. The germline BRCA testing done on patients identified 13% of patients with germline BRCA mutation. So this group only had 39 randomized patients and reached only about 60% of its projected accrual. So efficacy analysis in this group is quite limited because of that. Really, the group of interest for us for this trial was the BRCA-like group. BRCA-like testing on patients with germline BRCA negative status assigned 99 or 47% of patients in the BRCA-like group and the remainder in the non-BRCA-like group. When we look carefully at the qualifying biomarkers in the BRCA-like group, almost 80% qualified based on the HRD score, 17% based on BRCA1 promoter methylation, 7% based on germline mutations in other genes. We did have 8% that had somatic BRCA1 or 2 mutation, but they all had an HRD score of greater than 42. Because of lack of tissue submission and assay failure or lack of blood, we do also have this unclassified group of 75 patients. Moving on to the results, in the BRCA-like group, there was improvement in progression-free survival with the addition of veliparib to cisplatin with a hazard ratio of 0.53. Numerically, there was improvement in response rate, but this difference was not statistically significant. In terms of overall survival, we did see a trend towards improvement in overall survival with a hazard ratio of 0.63. There was no improvement in outcomes with the addition of veliparib in the non-BRCA-like group and unclassified group, and this is in line with a priori study hypothesis. We did not expect the PARP inhibitor to work in this group. We also did some exploratory subgroup analysis looking at patients who received this treatment in first-line setting. And here we see that there is consistent improvement in progression-free survival with veliparib compared to placebo with 2-year progression-free survival. I'm having trouble seeing this slide. Do you mind converting this into a full view instead of a presenter view? Perfect, thank you. With a 12-month progression-free survival of 3% in the placebo arm compared to 23% in the veliparib arm. Similar improvement in overall survival was seen with a hazard ratio of 0.53 and a 2-year overall survival of 20% in the control arm compared to 43% in the placebo arm. And note with the background that the average overall survival in these patients is about a year with standard treatment. One of the translational endpoints of this study was to look at the genomic instability score as a single factor to identify patients and place them in the BRCA-like group. So this subgroup analysis includes 75 out of the 99 patients that were in the BRCA-like group to see if we use only one marker, can we still demonstrate the efficacy of veliparib, and we see that we can. Patients identified based on just this score seem to benefit from veliparib with an improvement in progression-free survival. So in conclusion, what the trial showed that in germline BRCA mutation-negative metastatic TNBC, addition of veliparib to cisplatin improves progression-free survival by 47% and shows a trend towards improvement in overall survival. Half of the germline BRCA mutation-negative TNBC demonstrate a BRCA-like phenotype. So a very large chunk of these patients where a novel therapy can make a difference. And 2/3 of these patients were identified by an HRD score. 1416 cast a wide net using multiple biomarkers, but knowing that that's not really practical as we move this forward, we have to choose and narrow it down to 1 or 2 markers. The addition of veliparib to cisplatin was not beneficial in patients with metastatic non-BRCA-like phenotype TNBC. And really, this is the first randomized study in metastatic breast cancer to show the efficacy of PARP inhibitor in combination with platinum chemotherapy in biomarker-selected TNBC beyond those with germline or somatic BRCA mutation. There's many ongoing analysis. There has been a change in the landscape of metastatic triple-negative breast cancer since the conduct and completion of 1416. Now, immunotherapy combined with chemotherapy is standard of care for patients who receive first-line therapy and have PD-L1 positive disease. So, of course, assessment of PD-L1 on patients that enrolled on this study is very important, and also to look at the overlap between the BRCA-like phenotype and PD-L1 positivity. So this work is currently ongoing at KU under supervision of Drs. Godwin and Madden, and we'll be assessing if the veliparib benefit that we saw in BRCA-like phenotype is maintained when we adjust for PD-L1 status. The other exciting aspect of ongoing work is the integrated liquid biopsy work, which is evaluating two components of liquid biopsy for measures of HRD and to look if they correlate with benefit from veliparib therapy. And these include CTC HRD status and ctDNA homologous recombination repair mutations. The HRD status is being done using high-definition single-cell analysis at USC under the leadership of Peter Kuhn and Jim Hicks in collaboration with Epic Biosciences. This figure here denotes the flow pattern for HD-SCA with specifics for CTC detection, imaging, and large-scale transition scoring. CTC with 9 or more LSTs will be scored as LST high CTC, and samples that have 3 or more LST high CTCs per ml of blood will be denoted as CTC HRD positive, and some preliminary work suggests that 75% of pretreatment samples do have these numbers of CTC, and we're seeing CTC HRD positivity in almost 45% of patients, so I'm really excited about the completion of this analysis. So shifting gears just a little bit, to demonstrate that the correlation of HRD with treatment response may not be as simple as we think, and its interaction and overlap with immune upregulation may be very important. So this is work on an old SWOG study, 9313, led by my colleague Shane Steckline, that showed that immune activation and HRD have incomplete overlap in triple-negative breast cancer, and the different molecular subtypes within TNBC exist at variable proportions in the subclasses if we subdivide this cancer by HRD and immune upregulation status. And more importantly, this dual classification is highly prognostic in patients that receive DNA-damaging chemotherapy. Tumors that have both HRD and immune upregulation do the best. Tumors that lack both do the worst, but the ones that have one or the other are somewhere in between, suggesting that we can probably capitalize on absence and presence of one or the other for therapeutic gains. So I'll end with some concluding thoughts. There are many ongoing studies that are trying to target HRD in triple-negative breast cancer using combinatorial approaches. There are also agents in study that are looking at ways to induce or increase HRD, like with BET inhibitors or HDAC inhibitors. Liquid biopsy HRD biomarkers are being investigated, and we all understand the challenges of tissue-based biomarkers, so assessment of liquid biopsy samples here is very important. And finally, interface and overlap between HRD and immune infiltration is important and can be explored for therapeutic advantage. I want to thank all the patients that participated in this study, our many funding agencies, including NCI and the Hope Foundation, collaborating groups that helped complete the enrollment in the study, and the advocate committee. I'd be happy to take questions.
Great. Hello? Okay. Hello. Okay, that's better. Wonderful presentation. A ton of work. Can you tell me which part of that work was supported by the Hope Foundation, as this is the Hope Foundation part of the TM?
So the liquid biopsies work is supported by the Hope Foundation.
And that was going to be my question. In the BRCA-like group,
yes,
can you clear the BRCA-like phenotype in the circulation, and does it come back when you have a recurrence?
So that's exactly the type of question we want to ask. So since we don't have sequential tissues, but we have sequential liquid biopsy samples, this is exactly what we would like to do, look at the baseline and the cycle 2 and at progression samples to see what happens in those samples and correlate that with the veliparib response. There's decent data in prostate cancer that can be done, and that does relate to PARP inhibition response.
Well, that's a great introduction to David. We have time for one more question. And we do have a chat box, but I don't see anything in the chat box. So to stay on time, I thank you for the wonderful work, and we thank the Hope Foundation likewise for the continuous support of wonderful projects like this. Thank you very much.
Thank you.
So it's my pleasure to introduce David Mcconkey, who is currently at the Johns Hopkins University. It's hard to believe that David and I have known each other for nearly 30 years, as we're both in our mid-30s right now. I don't know how that happened. David is director of the Johns Hopkins Greenberg Bladder Cancer Institute. He's the inaugural director of this institute and professor in the Department of Urology at Johns Hopkins. And his mission, I'll just sum it up, is to make a difference by training people, by doing outstanding science, and by working with SWOG and the resources that we have. So David, great to have you here, and we look forward to your talk.
Great to see you again. Great to see everybody. It's great to be back. So I'm going to really focus, not surprisingly, on bladder cancer today. We've got great stuff in all 3 GU malignancies that we focus on, but we don't have time to talk about it all. So the other thing is that this is really team science and involves investigators at other cooperative groups, and so I'd ask you not to post anything and keep this confidential. There's unpublished work in here that I can't let get released quite yet. It's exciting. So many of us, as you know, were involved in TCGA projects, and these data sets have been disruptive. We still see almost every genomics paper uses them. They're characterized by really good genomic data, but not such good clinical data. And so our kind of overall vision is to try to combine the really robust clinical data that we capture within our prospective clinical trials with high-quality genomics and create resources for other investigators to do the kinds of discoveries that have been made with TCGA data. So overall, we want to digitize our cohorts, and this is a moving target. We want to get into some technologies that are going to be around 5 years from now. We don't know how the sample processing or what kinds of slides you're going to need. So this is a challenge. So to a certain extent, we do everything we can with the tissue that we have. So the second trial I wanted to highlight is the one we've got teed up now. This is Rob Svatek's S1602 clinical trial of one of the first immunotherapies. This is the tuberculosis vaccine BCG, delivered inside the bladder. We've been using it for a very long time. We still don't have a concrete handle on how it works. Rob's trial was designed to test the hypothesis that vaccination with BCG and PBD conversion would increase benefit from BCG within the bladder. It was also designed to test whether there was equipoise between the major source of BCG in North America, TICE, with a new strain called Tokyo. And so this trial not only addressed an important scientific question, it also addressed a really important unmet clinical need, because BCG has been subject to major shortages in North America. And this trial actually provided BCG for many of the patients at Johns Hopkins who wouldn't have gotten it otherwise. So the proposed genomic studies are the similar studies we've been doing on FFPE for a while. We want to do whole transcriptome RNA-seq, whole exome DNA sequencing, to look at biomarkers that may be associated with response and benefit. We would like to do some new stuff with the liquid biopsies, so not just plasma, but in GU malignancies, the urine is a rich source of biomarkers, and especially when we're talking about local disease, clearance of MRD, we really need a liquid biopsy biomarker in urine to be able to measure that. We have other things built in here too, including exosomes, a lot of rich material for future discovery. So there is actually pretty good prognostic data implicating some of the same biomarkers that you've seen in patients treated with checkpoint inhibitors. So high TMB is associated with good response. ERCC2, our friend, is associated with better outcomes. ARID1A and cyclin E amplification are associated with worse outcomes. So the biomarkers that we're proposing to do here are familiar to a lot of you, I'm sure, because of your interest in immunotherapy. Again, here's another paper showing that TMB status is associated with benefit from BCG. And even more data showing, again, that ARID1A in 2 different studies, so it's reproducible, was associated with worse outcomes when it's mutant. This is a study that was done by our friends at Memorial Sloan Kettering, and again, you can see this is immune signatures, and you can see immune hot tumors like you've seen with immunotherapy, immune checkpoint blockade, associated with better recurrence-free survival. So we've also teed up through a Team Translational Science Award that is led by Josh, DoD grant. We've also teed up our own discovery cohort consisting of about 150 tumors collected at Johns Hopkins. Woon-Young Choi was in charge of the RNA sequencing for this work. What's interesting, again, is that if you look at different stages of disease, it appears that we have different rules for BCG benefit. So in TA tumors, they look uniformly cold, and this may be consistent with their assignment to the luminal papillary molecular subtype as determined by TCGA. On the other hand, carcinoma in situ seems to be universally hot. In the middle, you can see the high-grade T1 tumors where we see this distinction between responders in blue and non-responders in red, where the responders are associated with immune hot phenotype. So this is also true when you look at the typical gene signatures you're all familiar with. So whether you look at CD8 T cell signatures, interferon gamma, we've looked at B cell signatures. The T1 tumors are distinguished by the responders having hotter tumors by all these criteria, again, that you're all familiar with. So what we'd like to do, and we're right now, Josh and Rob and I are in active discussions with a lot of different groups to try to figure out how we do this, is to have a really accurate MRD measurement for urine. Because when we treat patients with intravesical therapies, we're looking for recurrence. We're not looking at response as measured by reductions in tumor volume. And so having a really good MRD measurement would be, I think, very important to interpreting our results. Here we have a result that was just put online by my colleague Lars Dierskot, who's a leader in liquid biopsies based in Denmark. And using an Atera-like assay, you can see here that the patients who cleared their tumor DNA had better progression-free survival, or recurrence-free survival. It's not quite as robust as I would have hoped. And so I think there's probably room for improvement. I think we probably need a more sensitive MRD assay. And if you're coming to our oligometastatic meeting tomorrow morning, we'll have more discussion around that. Finally, on the horizon, very exciting. This is a very disruptive phase 3 clinical trial led by Parminder Singh in our group. This is loaded with biomarkers as well. And importantly, it addresses a really important emerging unmet need in our bladder cancer community, and that is bladder preservation strategies. Thanks to a very strong relationship that we have, our committee with Genentech, we've got very strong input and support from them in many of our different trials, and this is certainly one that they're playing leadership role in designing the translational medicine studies. This is about 50% accrued. We think it'll be about 75% accrued within about 6 to 8 months. Parminder probably has a better idea of that. And then we'll really be locking down our plans to study the translational medicine biomarkers in this. So here are the collections. Again, you can see we're getting blood, tissue, and urine from this trial. We've amended the protocol to get blood and urine at later time points, 1 year and 2 year time points. We really are excited about this trial. So this is another local therapy trial like BCG. And so we're really interested in bladder preservation and making sure we eradicate local disease. We'll be able to measure if there's any subclinical metastatic disease with the ctDNA measurements in the plasma. And again, we've got immunotherapy on board here, so it's not just about measuring biomarkers for radiation therapy, but it's also rich in biomarkers for immunotherapy. So I just wanted to highlight the large team of investigators that performed the Coxon trial, first off with Tom Flagg, and then Kathy Tangen and her team at Stats have been crazy busy, not just with this trial, but with every other one. And they deserve a serious shout out for all the work that they do for us. And finally, to my close collaborator, Woon-Young Choi, first at MD Anderson and now at Johns Hopkins, who really made sure that the DNA and the RNA that we got out of these tumors was high quality and satisfactory for the analyses that were performed at Memorial Sloan Kettering. So with that, I'm very excited to share this with you for the first time. Again, please keep it quiet, and happy to take any questions.
David, thank you. You were within 10 seconds of your target time limit, so I am, and on the right side of it too. So
I know you very well.
Any questions from the audience? And I'm looking at the chat box. I don't see anything in the chat box yet. So I'll ask, you know, being a GI person and hearing about urine ctDNA, or not ctDNA, but urine DNA, measuring this, is this different tubes that you need, different preps, different storage? Tell us a little bit about the techniques for looking at urine.
So one of the decisions we're all making now is whether to continue doing it ourselves or to bring in companies to do this. And so one of our former committee members, Teresa Copey, many years ago began a partnership with Illumina to establish a liquid biopsy in urine. They have a company now called Convergent that has, I guess, 40 or 45 employees, and they have what looks like a very robust urine tumor DNA measurement platform. And again, because Teresa's been involved in that work, she's concerned with all the same MRD questions the rest of us are concerned with. So I don't know if we're going to be able to tell you that answer very clearly. What we got is what we got. So for the 1806 trial and for the 1602 trial, the urine was collected using, standard techniques, urine sediment collections. We don't know if it's going to be optimal for Convergent. And if so, we may have to adapt the technique to suit the material we have. Going forward, it'll be about nominating the right platform and company, and we don't know if it's going to be, a Signatera-like assay or a methylation-based assay. There's a lot of promise with those, or a whole genome assay. So we're all going to have to keep talking about this, I think.
It's evolving.
Fred Hirsch, Mount Sinai. I come from another community too. I'm a lung cancer person.
I know, yeah.
So, in lung cancer, we are struggling with the TMB concept. And we are struggling with lack of harmonization of the assays, different assays used, different cutoffs used. And personally, I don't think all mutations are born equal in terms of immunogenicity. So my question, I see bladder cancer are using the TMB, and it looks like it comes out as a promising biomarker. What is your, you don't struggle with the same issues as we do?
So again, I think if Tom Powell's were here, he'd probably be saying a similar thing. None of our biomarkers for immunotherapy is very good, really, realistically.
Yeah.
I think, are plagued by a lot of problems. So one thing is sampling. A lot of our clinical trials we're sampling tumors that are distant from the lesions we're actually treating in the patients. The TURBTs, the biopsies that we're sequencing, really, they're out of the patient already. They may not look like the tumors that's left behind. We're starting to see direct evidence for that in some of the work we're doing. The other thing is the dynamic changes, right? So we're taking snapshots before therapy, and Pam Sharma and others have shown, Jennifer Wargo, that things change when you put pressure on the immune system in ways that may be even more predictive than the baseline measurements. So we don't think TMB is going to change clinical practice, but we do think we need to sequence these tumors.
I totally agree.
Yeah.
Yeah, Fred, I was going to ask you for your opinion, but we got it. So, yeah. So, David, in my first question, you described you answered it about the methodology, but how do you collect urine and store it? for
So-
later use with the proper assay.
So we have commercial preservatives that we can add to the urine, and a lot of that has been done in trial of adenoviral interferon that you're probably familiar with because Colin Denny has been involved with that quite extensively. But typically with these multi-center trials, the handling of the tissue in the urine is a bit hit or miss. So with 1602, we got what we got. With 1806, we've started to talk about additives. I think as we start to launch new trials, we're going to have to lock down an SOP. And if it's not perfect, at least we'll know that everything's the same.
So still an evolution.
Yeah, it's still an evolution. Yeah.
Okay.
And urine's tough. We're using Streck tubes for everything, but urine is tough. So.
Okay. Thank you, David.
Thanks,
and
Lee.
Thank you for the participation. And our last speaker in Plenary I is Ignacio Wistuba from MD Anderson Cancer Center. Ignacio's division head at Interim Division of Pathology Lab Medicine. Seems you've been that for a long, long time. And professor of the Department of Thoracic and Head and Neck Medical Oncology. He's co-director of the Division of Pathology and Laboratory Medicine. And similar to my other friends, we're longtime friends, but I haven't seen Ignacio in Houston. So the last time I saw him was out here at the SWOG meeting. So it's good to see you again.
Thank you. Thank you. Thank you very much, Lee and the organizing committee for the invitation to be here. It's a real pleasure to see you all and many friends that I missed for 2 years. And today I feel that I'm coming to report back on what is the status of the cancer immune monitoring and analysis for cancer network called CIMAC. Some people call CIMAC, but I call it CIMAC, that work closely with Data Commons Center that is called CIDC. And this is part of the NCI Moonshot program to develop biomarkers for immunotherapy in cancer. And I presented, I believe, 3 years ago when we were starting this network. And today, I'm going to show some of the mission and strategy of the network, what we have done in terms of assays and platforms for biomarker discovery and correlative studies, some of our accomplishments, and then I'm going to highlight some collaboration between our CIMAC network and SWOG, and then some few lessons learned and future work of this very ambitious network. So this is actually an initial slide showing the interaction of this and the composition of the CIMAC CIDC network. We have 4 sites, Stanford, the PI is Holden Maker, Dana-Farber, the PI, contact PI is Kathy Gu, and from Mount Sinai, Sasha Nyatik, and from the CIDC is Ethan Serrani, and I'm the contact PI for the MD Anderson CIMAC. So we have become a very strong collaborative and collegial community of investigators that we all together, when we talk about the progress of our network, is over 100 people that actually doing work. And the purpose is to work with clinical trials on immunotherapy that had collected specimen as part of the NCI clinical trial network. But we have actually a friend invited to the party, which is in a yellow figure there. It's called PACT, which is a partnership for accelerating cancer therapy that is run by the foundation of NIH, and it's a way to bring trials to our network funded by pharma. And they come and take, and participate in our common effort. As was published in Clinical Cancer Research a year ago, these are the main objectives of the network. I'm not going to go through these, but the idea is to do correlative and exploratory studies, develop a standard SOP for biomarker analysis, immunotherapy, bring a group of multidisciplinary investigators in the field of biomarker development, and hopefully bring as much innovation as possible to this type of work. Again, a more detailed kind of structure of the network with clinical trial coming from the NCI and the PACT group that come to our group of clinical working group to assess the quality of the trial and the correlative study proposed, and then we all make a decision on how to distribute the biomarker analysis, some in which we have a lot of collaboration and exchange of specimen between all the 4 CIMAC sites. And then the analysis is done at local CIMAC in collaboration with the group statisticians, which is very important, and then at the end with the CIDC that actually provide additional bioinformatics or statistics support, and they are developing a common database for all these trials, for all the biomarkers that we're doing, that in the future hopefully help us to do inter-trial biomarker exploratory analysis. We all want biomarkers analysis in the context of immunotherapy being comprehensive, hopefully longitudinal, something extremely hard to do in the context of a clinical trial group like SWOG or others, especially when we are trying to get tissue to do this type of analysis. And in the context of immunotherapy, not only tumor matters, you all know that we have also to think about the host, the patient. So we have taken a large number of biomarker approaches to study, to interrogate tumor tissues, blood, and fluid from patients, and also we are exploring the microbiome. And this is just a list of platform and assay that are at our disposal. I'm going to talk about them a little bit more in the next few slides. So one thing that we did first is to develop what we call an umbrella protocol that actually is available for everyone in the NCI network, which is to establish a consistent process for the site and biorepositories involved to collect and process samples for biomarker analysis in the context of immunotherapy. This standard operation procedure has been already published, as I said, and is available actually in this website that we maintain for our CIMAC network. Then we spend a lot of time on deciding what's going to be the biomarker that we're going to use for all trials. And we came with a tier system for biomarker approach, tier 1 and tier 1 alternative, in case that some of the samples were not good enough for the tier 1. We have some alternative to do analysis. For example, if we don't have good quality for RNA sequencing from formalin fixed paraffin embedded tissue, we can run nanochain or for RNA analysis. So the tier 1 for all or most trials consists of whole exome sequencing, multiplex immunohistochemistry or immunofluorescence analysis, and PD-L1 immunohistochemistry from a tissue, formalin fixed paraffin embedded, most of them, right? And RNA-seq. Then we are the option to do TCR sequencing from the tissue and blood specimens. We develop a panel for cytoph, and if not possible, flow cytometry from blood specimens. For cytokines and growth factor analysis, we have access to the Olink platform, if not Luminex, and ELISA in some specific markers. And for an alternative to whole exome sequencing, we also thought that having cell-free DNA NGS panel analysis could be important, especially when we don't have normal tissues or sample from the patient to run whole exome sequencing. And then we are added microbiome. We have tier 2 samples in biomarkers, that are more advanced, potentially other multiplex analysis of immune infiltration in tissue like MIVI. Now we're thinking about Codex or other things like that. And then we have tier 3 that are highly novel and exploratory biomarkers that we are adding on a limited number of trials in which we have a prospective collection of specimens. The second thing after the third thing was to develop an SOP for the sample collection processing that's available now for prospective collection, then decide the biomarker approach for all or most trials, and then we spend 1 year and a half, maybe 2 years on harmonization of assays. So the tier 1 assays were harmonized, and these were published in 3 publications, along with the one described in the network, in Clinical Cancer Research as a package in 2021. And I'm going to go very briefly about what we have done as a team. I'm here representing about 100 people that have worked on this. I shouldn't take credit for any of these. The only thing that I've been doing is helping to chair the network and be one of the PIs at MD Anderson. But we started with site of harmonization. We designed a study in which the four sites participated with pre-stained and pre-stained samples in one site, or stained samples, PBMCs, right, in each site, and we find a very high correlation for a large palette of site of marking with a very strategic gating system to identify subpopulation of immune cells. So we show that site-of can be standardized and harmonized with coefficient variants that are at least equivalent to what we have seen in the world of flow cytometry for immune cells characterization. Then we harmonize the multiplex immunohistochemistry and immunofluorescence analysis. So we use 2 platforms. For multiplex immunofluorescence, we use a panel using the Vectra Polaris system. And for multiplex immunohistochemistry, we use chromogenic consecutive staining on a single slide. And we're able to actually show using whole section and tissue microarray from various samples and tumor, including head and neck, breast cancer, and lung cancer, that it was a good correlation for key biomarkers on the analysis of tissues, like CD3, CD8, PD1, PD-L1, cytokeratin, and between 3 sites that participate in this study, 2 using multiplex immunofluorescence and 1 using consecutive immunohistochemistry staining, with a very good correlation between the analysis of this biomarker, despite we were analyzing consecutive sections of the tumor specimen. So we show that it's a comparable quantification interpretation of this assay despite different instrument platform clones and operators and analysis software. Then we pursue the harmonization of whole exome sequencing and RNA sequencing using initially a pool of cell lines DNA, and then we move to tumor tissue, formalin fixed and fresh frozen coming from lung cancer. 3 sites participating in this, 2 CIMAC, and we invited MOCA, which is an NCI genomic lab with experience on whole exome sequencing and RNA sequencing to have more data to compare. And we show actually in whole exome sequencing that we had a very high level of concordance on the target region between the 3 centers in the whole exome sequencing, and also a very high level of concordance on calling mutations in non-small cell lung cancer specimen that was tested. And we chose lung cancer because it's a tumor that had a high level of genomic abnormalities, and I'm a lung cancer researcher, so I have them available to do all this work. And this actually shows more detailed analysis on the whole exome sequencing and RNA-seq, in which we develop very clear criteria of quality control on the quality of the RNA that we extracted from formalin-fixed paraffin-embedded tissue for RNA-seq using different criteria in terms of transcriptome quality data. So where we are at now, I'm showing data from 2021. We have our last group meeting in which we report at that time that we have 33 trials being analyzed by our network. As you can see, there are 26 more that are planned or we are in the process of adopting in our group. And we expect to study overall sample for more than 3,700 patients. And the clinical setting varies a lot, as also the modality of the clinical trial. And you can see there is a relatively higher number of immune checkpoint inhibitors trials. We have some cell therapy, and we have some combinatorial approaches too. And this is a slide that actually shows the number of clinical trials using each of the at least tier 1 assays. And you can see there is not 100% consistency on the assay that were run per trial, and you know why. Availability of specimen. And this is the big challenge that we're facing on samples that have been already collected as part of the NCI clinical trial network. The quality and the quantity is not what we need for this type of approaches. We are getting data, but we think that we're going to do better when the trials that have been collecting prospectively using our umbrella protocol since we started the network. However, having a panel of markers or the assays as we are doing that actually overlap in multiple trials give us an opportunity to do inter-trials analysis, and we're planning this. So briefly, I would like to now show the experience and preliminary data, work in progress that is happening with the SWOG group that I'm very happy to report. And these have been led mostly by the MD Anderson CMAC. So the first is some data that we have been collecting in collaboration with the wonderful clinical investigator by statistical support from the DART trial, S1609. I would like to acknowledge Dr. Sandeep Patel, John Kwang Che, and Rachel Kuzrock for their partnership. And this is a trial that you know well. It's focused on rare tumor using a combination of anti-PD-1 and CTLA-4. And with the treatment that is shown there. And then they have for these trials some of the cohort of rare tumor, baseline tumor tissue, blood collected at different time points, and then they had planned to do some biomarker analysis. So with working, and these are some of the data that I have shown for the neuroendocrine tumor cohorts, the one that I'm going to show some data today, that they have a significant response to this type of therapy that go from 22% to 44%, especially if you look at the high-grade neuroendocrine tumors. So in our first analysis, we got only 37 samples from 3 cohorts that are neuroendocrine tumors. We propose the hypothesis that our exploratory analysis could hopefully identify biomarkers that are associated with response or non-response to this type of therapy. And we developed a biomarker plan in which we were intending to do a multiplex immunofluorescence analysis to study the immune infiltration of the tumor, and NanoString because the material was not quality enough to do RNA sequencing, so we ran a panel of 770 IO genes. And also we have plasma PBMCs collected for cytoph and cytokine analysis using the Olink. I'm going to show briefly some of the data that we have obtained in this neuroendocrine tumor cohort. First on the a study of 730 immune-related genes using RNA gene expression analysis by NanoString. We have identified, as you can see in the heat map, clearly a different signature, one of them that is associated to the few responders that we have, and there is some specific genes associated in this immune-related signature that show a clear difference in the level of expression between responder and non-responders. So we have found that the gene expression profiling showed an enrichment of immune-inflamed tumors in the responder in these neuroendocrine cohorts. So in terms of analysis for immune cells, we have used the NanoString data using the timer analysis to estimate the infiltration of some subpopulation of immune cells, and we have found that the infiltration of myeloid dendritic cells and B cells also in this population correlated with a higher expression in the responder group. And then also we have some specific data on the immune infiltration done by multiplex immunofluorescence, and we have found that cytotoxic T cells, Treg cells, and exhausted CD8 T cells level of infiltration also associated with response. These are all data that are coming from the analysis that we're trying to make sense of it. It's very complex, but some of them seems very interesting. And as a conclusion from this type of analysis, both gene expression and immune cell infiltration done by nanostring and multiplex immunofluorescence confirmed a higher level of specific population of immune cells associated with tumor responders in this population. So as a summary, I'm going to skip some of the slides for sake of time. On this early data showing that in this neuroendocrine tumor cohort of the DART trial, activated immune presence in the tumor tissue at baseline correlate with improved patient's outcome. We have found that myeloidentitic and B cells may play a response in this population to the therapy that was given to these patients. PD-1 and PD-1 expression, actually, they did not show correlation with response, and this data have not been shown. And we are now also looking at the site-off and all-in data, and we have some preliminary data that maybe the expansion of memory and activated CD4 T cells in the periphery, in the blood, correlate with response, but I'm not showing those data. The next step is take the multiplex immunofluorescence analysis and do spatial analysis to look at not only the density of immune cells, but the location and how they relate to each other and to the malignant cells. And we are expanding the analysis to TCR sequencing and continue working with the wonderful clinical collaborators to actually come up with solid results that hopefully we can validate in other cohorts. And then briefly, I'm going to talk about some of the preliminary data that we have obtained working with the wonderful group of lung map investigators. We have been studying the S1400I cohort of this phase 3 randomized clinical trial that has been published. Is a study in this cohort studying the effect of adding ipilimumab to the nivolumab treatment in patients with stage 4 squamous cell lung cancer previously treated. And this study actually was published. The adding ipi to the nivo regimen actually didn't show any difference in terms of survival to the patient. So what we have been doing in the studies, again, coming with very exploratory hypotheses, and we have here a very ambitious biomarker plan. We have a tumor tissue available at baseline. We have been doing multiplex immunofluorescence. These are 14 markers, 2 panels using vector polarities. We have done nanostring RNA expression analysis, whole exome sequencing, and from plasma specimen, we are doing cytokine all-link testing. You can see that from the multiplex to the whole exome sequencing, the number of samples available are dropping, which is, again, show the challenges to work with these already banked specimens collected. We have time to event, clinical endpoints, response rate, overall survival, progression-free survival. And here, this is very important to show that only 36 patients actually have complete biomarker panels. Very challenging, right, to come up with solid data, but we think that we can come up with some provocative hypotheses. If you want to see more about this, please come to the Lung Cancer Committee on Saturday. I'm going to talk about S1400I analysis, but I would like to highlight one thing for you that I think that is very important in terms of biomarker analysis when we study the immune infiltration of tumor and particular VNA pathologies. This is a platform that we have used at the MD Anderson and other centers. I already mentioned about this multiplex immunofluorescence. You can combine up to 9 markers, do a quantification, but also you can do a spatial analysis looking at the relation between the cells that you're studying. And you can study stroma and tumor. You can come up with phenotypes you predefined. You can come up with phenotypes that you haven't predefined. In this case, we predefined 17 phenotypes that we're able to study in 82 tumors in this cohort, 35 treated with a combination of immune checkpoint inhibitors, 47 with a single markers. And here we can see actually the data by arm And I would like to highlight the fact that in the Nivo arm, only single treatment, single agent treatment, we have that memory or memory-related T cells, actually the higher expression of these cells in the tumor associated with better outcome in terms of PFS or overall survival. Very interesting observation that we would like to take and study in other clinical trials using this type of therapy. And also for the combination, and this has been actually validated in the multivariate analysis, that the presence of regulatory T cells, higher density, actually associated with lower overall survival, that makes sense from the immunologic point of view. But also we have taken this analysis and done spatial analysis, studying the distance of all these phenotypes. And we have found, for example, in the Nivo plus IPI arm, that a closer distance of cytotoxic T cell activated to malignant cells staying with cytokeratin associated with better overall survival. And then if those cells, the malignant cells, PD-L1, the presence of cytotoxic T cells closer also associated with progression-free survival. That also makes sense from the perspective of the interaction in between all these cells. So lesson learned, probably I could talk about 2 hours about what we have learned putting this network together. But I think that it's important to highlight the advantage of collaborative research, team science, and they have the opportunity to actually to share expertise and being together in a community. You actually share a lot more than usual classic collaboration because you are in the same boat. You want the network to be successful, to continue, to hopefully be renewed, right? And then I think that we need to improve the interaction with our own group banks, biorepositories. I'm a PI of the EcoAcri Bank. I work closely with Jimmy and others for the SWOG Bank, and we know that it's very challenging actually to be able to collect their best samples for this type of analysis, and then being to interact with a new network that actually have 4 laboratories and they have their own kind of goals, right? But I think that we have done a good job there. Of course, the need for harmonization of assay protocols in view of the data variability. I think that our harmonization studies are very important on this. The importance of the statistical considerations. I have collaboration with a good statistical team, and in terms of SWOG, we have been actually very fortunate to have that collaboration, and we appreciate that. And also to be flexible, because the biomarker and platform that we selected 3 years ago may not be the best today, right? For example, multiplex immunofluorescence, 9 markers. We can do codex, 40 markers, or maybe 40 markers, right? So we need to adapt. We need to move from whole exome sequencing to single cell sequencing, right? But we need the right specimen for that. And also the importance of administrative support in all fronts. This is really require a village to be able to deal with the amount of paperwork that we need to handle. In terms of one trial require 5, 6 MTAs. And you can imagine what that means, right? So with that, I would like just to show you as an example, one of the team, the Anderson team on the CMAX, a large group of people, and each of our sites actually have a similar number of collaborators. Some resources on the CMAX ADC network that you can look at, and I'm happy to share most of these slides, so you can check yourself, and thank you very, very much for your attention, and happy to answer any question if I have time.
Well, very simple to understand, thank you. Fred.
I didn't get that irony. I mean, I've been doing Zoom so much that I cannot read body language now anymore.
Thank you, Ignacio. That was, as usual, very elegant. A couple of questions. You pointed out the quality of material several times. And, CMAC has existed for a few years. I wonder if we are using CMAC in a wrong way. We shouldn't use them for retrospective analysis, but we need to include CMAC mechanism much more in prospective analysis. And to that end, does CMAC have SOPs? They do.
Yes.
So, you know, we should include them much more in prospective studies.
Yes.
Yeah.
The challenge was to actually put together a network that we all were happy to be awarded, but our colleague from NCI had no plan actually how it's going to run. So we have to build this from scratch. That took us probably a year and a half. Then we had to define the biomarkers and the platforms and validate them in some sort of harmonization, you know, work that took another year and a half or two. And it was very painful to do, you know, harmonize this assay that we all run in our lab, but now need to actually expose and do this comparison with others. And actually we call the H word, the harmonization, because we don't want to talk about it anymore. And third is to get right samples to answer some interesting questions. And the only way actually to show data today, 4 years after we started this network, is to go to retrospective collected specimens. So we call those hybrid studies. So studies that, you know, we're doing analysis, but we shouldn't be doing, but we're doing because we needed to start. And I think that probably by the end of this year, early next year, we will see data coming from studies in which the CMAC has participated in the design of the collection of the specimen using our protocol. The question is that in a year from now, officially the network ends, right? So, and that's a question that NCI needs to resolve because we are just getting there and then our first 5 years is ending. I hope it's going to be renewed. As an investigator, we always get hope to continue being funded, but I really don't mind who is going to be doing this work. I think this work should continue based on what has been done so far. Sorry for the long, politically charged answer.
No, it was filled with emotion. That was great. Heinz.
Heinz Josef Lenz, USC. So this is an incredible effort. Anybody who is involved in this large collaborative grants only has a glimpse how difficult and how much work it is. Now, I think we are all understanding tumor heterogeneity as a big challenge of anti-cancer treatment. What about the heterogeneity in the tumor microenvironment? There are significant differences, not only from the site of the location of the primary tumor, but metastatic sites from one cancer to the other. How do you going to address this? You already have the challenge of specimens. You don't even know, sometimes you have only the primary tumor of a liver met or lung met. How do you going to face and address these challenges in the future with this incredible technology?
Yeah. It's a great question and have a very different, it's a very difficult to answer that. The way I see this is as a biomarker researcher and a pathologist, I deal with the sample that I can get, right? In our institution, we have a longitudinal collection program called Apollo. It's part of the MD Anderson Moonshot program. We have longitudinal collection of biopsies in 2,000 patients. We can study longitudinal evolution of the tumor microenvironment. We have multiple site collection. In some cases, we can study heterogeneity, but that's not possible in most of the trials that we are dealing with, right? So we need to actually use what we have. The way I see this, and I'm learning immunology, and it's very hard, right? Is I see kind of a parallel what we did when we studied genomic of the tumor. We were all worried about heterogeneity, not only at DNA level, but expression and also at protein level, right? So the way I see this is in response to immunotherapy or the immune response to the tumor, we need to access to the tumor to understand what's happening there. Whatever sample you get, study all what you can to understand the playbook of the immune response to the tumor with or without intervention. And then, if you identify a signal there, you can take it to the blood or the fluid and see if you can follow it up and understand the cause of heterogeneity. In genomic, we're at that level, right? We study whole exome of the tumor, whole exome sequence, whole genome sequencing of the tumor. We know what the tumor have in general, and then we go to the cell-free DNA, to the urine, to other fluids to study what we want to do in more detail. I hope that we can reach that level at some point in immunotherapy in which understanding what's happening in the tumor or in the lymph nodes, on the tertiary lymph node structure, then we can take that information and trying to look for those signals that are diluted in the blood, in the cells, or in the fluid. And then there have a better understanding of the heterogeneity.
Manuel Espinosa, Indiana University. Thank you for your talk. I'm more interested in malignant hematology, but something we've noticed in that field when we look at the tumor microenvironment is that sometimes humble cytotoxic chemotherapy has more impact on the microenvironment than targeted therapy does. I wanted to see, I know that you've mentioned, doing retrospective analysis on existing trials, if you're planning to look at trials that have used cytotoxic chemotherapy and see how that impacts the immune microenvironment.
It's not the primary focus of the CIMAC. We debated that. We were desperate on samples, so we wanted to actually study that. I think that it's very important field of research in biomarker development, but we have some studies in which there are combination between immune checkpoint inhibitors, some other immunotherapy approaches, and chemotherapy. It's going to be hard to actually dissect the effect of both interventions, but that's as close as we are getting in the chemotherapy or in the cytotoxic analysis. Thank you.
Yeah, and you'd have to have a pre and post for each patient. And
the answer is neoadjuvant studies, right? In any tumor, and David, what you have shown in bladder and the community in lung cancer actually is doing is you can have, these neoadjuvant arm, one immunotherapy drug combination, and then with or without chemotherapy, right? And then you can see the effect. I don't know if the effect after 2-3 cycles is enough, but at least you can start. And then you get entire tumor to play with when it's resected.
So one question online, which is a good way to wrap this up, because I will paraphrase this. Nicole Kuderer stated, such impressive and interdisciplinary efforts with limited tumor specimens, core biopsies, for example. What assays do you like to prioritize for IO response assessment? So I will ask you, what are your 3 favorite assays? What do you think are, if you only had money to do 3 assays when a patient comes in the door, they have metastatic disease, what 3 assays would you prioritize if you only had $100 million?
H&E staining, because I'm a pathologist, I want to make sure they have tumor. Then I would run a multiplex immunoprofiling strategy, Codex, maybe Hyperion IMC, or the NanoString DSP GeoMX. And third, RNA-seq.
Okay, you heard it. There it is.
Thank you.
With that, I think we'll wrap up the afternoon. Thank you. And thank all the presenters and all the folks who stood up and participated. And I'm happy to hear suggestions for the next plenary session that we have coming up in just 6 months. So if you have some ideas for great speakers or new topics, please email me directly. Otherwise, have a great, safe meeting, and it's great to be back together again. Thank you all.